After labeling the bacteria in different medium, they spin the bacteria in ultracentrifuge, and heavier bacteria with DNA will fall in the bottom of the test tube, and lighter DNA like will be at the top of the test tube. So basically, in zero generation, everything is 15N. So that's why it's heavier and it's in the bottom. In the first generation, because the DNA have two strand. One strand originally from the 15N, and one from 14N that was just made. It's only it's it 100% is found in between N15 and N14, and after that, um, the amount of N15 is the same. However, because there's an increase of M14 DNA strands, the ratio of the N15 is decreasing while M14 DNA strand is increasing.
Representation of what is happening to the bacteria in each generation. So in the original generation, you have bacteria with 100% heavy nitrogen, or N15. And then as they make copies of themselves in the N14 medium, the new template strand that is going to be added to the original DNA will contain N14. So now we have two hybrid DNA strands of N14 and N15 DNA. So in generation 2, these hybrid strands will split here, 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 and here, and the new template strand will be added to either DNA strand with N15 or N14. And as a result, there will be two hybrid DNA strands and two N14 strands. And then in generation 3, it does the same thing, creating two hybrid DNA strands and 6 N14 strands. And then in generation 4, it'll do the same thing, creating 14 N14 DNA strands and 12 hybrid DNA strands. So the more generations you have, the less you're going to see of N15 and more N14.
For the conservative model, we're going to start off with 100% of the heavy mixture, and then the next generation is going to have 50% of the heavy mixture, 50% of the light mixture, and then generation 2, 75% of the light mixture, and then 25% of the heavy. So the main thing to see here is that there's no mixing of the strands, which means that there's no hybrid molecules produced. It's always going to stay either N14 or N15, nothing in between. So on the bottom, we start off with the original heavy DNA molecule, and then after each generation, the heavy DNA molecule never really disappears, but it's quickly outnumbered by the newly synthesized molecules of light DNA, and you also see that there are no hybrid molecules, so the two strands of the molecules are always the same mixture.
In dispersive replication, every single new copy of DNA has parts of the original DNA and the newly replicated DNA. After every generation, while all copies of DNA have parts of the original and the new DNA, the amount of original DNA is reduced in the new copies from 1/2 to 1/4 to 1/8, etc. As for the experiment, since every strand of DNA has the same ratio of original to new DNA in a given generation, there would only be one band when undergoing centrifugation, since they would have the same densities and the same ratios of the new 14N to 15N. With each successive generation, the density lowers slightly because of the number of new 14N DNA increases, but it will continue to stay as one band.